Hello, my name is Andrei Skirtac. I'm leading the nanobiotechnology laboratory at Ghent University, and today we'll speak about IFM, Raman, and other type of microscopy methods for biocolloids and bacteria applications. Which other methods exist? Well, we look here and we see three major parts. The first part is fluorescence and electron microscopy. There is also Raman and atomic force microscopy modalities that are available for biocolloid and bacteria research. In the presentation, we'll start first with fluorescence and electron microscopy, shifting later to Raman and IFM. Fluorescent microscopy is a golden horse of microscopy in general. When we hear about microscopy, very often it is meant that fluorescent microscopy will be used. For biocolloids specifically, it can be different modalities can be applicable, where one can look at membranes, some enzymes on the surface, some molecules, and even ions. The colorful images that you can see on the right-hand side typically would identify some of the subcompartments and components that are that we are capable to visualize. Visualizing molecules is something that was done for a long time, but for example, in this slide, we can see that even ions, in this case, some reports were made that chloride ions were investigated in bacteria. Fluorescent microscopy has many different modalities, like, for example, time-resolved FRAP, which is fluorescent resonance after photobleaching studies, or time-resolved film, time-resolved signals in bacteria. They provide a wealth of information, and very often fluorescent microscopy is superimposed or presented next to electron microscopy images that you can see on the left-hand side. A lot of advantages of advanced microscopy include a wealth of information, as I mentioned earlier. This is a very versatile technique, and this is also a very sensitive technique. Combined with electron microscopy, it can also provide insights into fine structures of bacteria. But as the nature of the name suggests, that in fluorescent microscopy and even electron microscopy, labels are often used to visualize the samples. That's why some other type of microscopy are necessary. One type of microscopy that we would like to accentuate here is Raman microscopy. This is a label-free method in which scattering takes place of photons on the virtual level of molecules, and the energy difference between the initial and the scattered photons provides information about vibrational fingerprints of molecules in biomolecules. So on the right-hand side, one can see that different applications were presented. We'd like to mention that for typically for bacteria research, the spectra are quite complicated, so different techniques are used to decompose the images. For example, principal component analysis, or these days also machine learning analysis. And on the right-hand side, you can see one can see that, for example, for E. coli, different subpopulation of cells, it was possible to visualize different population of cells. What is very interesting is this next slide. This slide shows that it is even possible to find the difference between different phenotypes of bacteria, as it was reported just a couple of years ago. With Raman microscopy, bacteria identification is possible. One can also investigate antibiotic resistance. Extensive databases are available now to visualize, to identify biomolecules. And bacterial phenotypes are also, it was reported that bacterial phenotypes are possible to be distinguished. In addition to this label-free microscopy modality, one can also report here atomic force microscopy, which is often used to visualize topography of the samples. Some early research by Yves Dufray shows here that images of a high resolution of bacteria that was done some time ago. One can also, with this technique, with atomic force microscopy, one can also pull on the molecules, and by this unbinding of the molecules can be investigated by this sharp drops that you can see in the signal force versus extension distance that can be seen on this image. In application to cell biology, one can also distinguish with atomic force microscopy different regulated cell death modalities, and with atomic force microscopy it is possible to distinguish them much earlier than the first onset of fluorescent signal, as you can see in this slide. Interestingly, but atomic force microscopy can even provide information on the different forces that are involved in adhesion of bacteria to the surfaces, as can be inferred from this slide. To conclude the presentation, various techniques are possible to visualize biocolloids and bacteria. Fluorescent microscopy is still a golden horse, is a very powerful technique and very versatile tool with different modalities that it can offer, but it uses labels. So a number of label-free techniques are available. One of them is Raman microscopy, where vibrational fingerprints of biomolecules are used and can be applied for bacterial and biocolloid identification. Atomic force microscopy is used to visualize topography, but importantly, mechanical properties and adhesion of molecules and biocolloids can be probed. So full potential of these techniques is still yet to be realized, but extensive progress has been already obtained. Thank you for your attention.